Untuk kita semua, semoga Bapak, Ibu, dan Saudara dalam kondisi sehat selalu. Selamat bergabung di acara virtual workshop CAS Scifinder yang diselenggarakan oleh Perpustakaan Universitas Erlangga bersama dengan CAS Scifinder. Senang sekali saya Safety yang akan memandu acara hari ini. Saya ucapkan selamat pagi dan terima kasih atas kehadirannya. The Honorable Mrs. Pamela on Application Specialist Southeast Asia speaker today. Wiwi Wijayanti dari iGroup sebagai moderator. The Honorable Mr. Adran Lee as Customer Success Manager from CAS Scifinder. Yang terhormat para dekan fakultas di lingkungan Universitas Erlangga atau yang mewakili. Yang kami hormati Kepala Perpustakaan Universitas Erlangga. Yang kami hormati para dosen, peneliti, serta mahasiswa Universitas Erlangga. Baik, untuk acara hari ini meliputi sambutan, pengenalan OpenVPN, pemaparan database, sesi tanya jawab, dan terakhir adalah penutup. Baik, untuk acara yang pertama adalah opening speech yang akan disampaikan oleh Ibu Kepala Perpustakaan Universitas Erlangga, yaitu Ibu Suherni, SS MSI. Kami silakan.
Ya, matur nuwun Mbak Seti. Assalamualaikum warahmatullahi wabarakatuh. Selamat pagi dan salam sejahtera untuk kita semuanya. Yang terhormat para guru besar, dosen, peneliti, mahasiswa, Sivitas Akademika Universitas Erlangga, Miss Pamela On, tim iGroup, dan tim perpustakaan yang berbahagia. Marilah kita panjatkan puji syukur kehadirat Allah Subhanahu wa Ta'ala atas nikmat dan hidayahnya, sehingga pada hari ini kita bisa diberikan kesehatan untuk dapat mengikuti acara virtual workshop CAS Scifinder sembari menunaikan ibadah puasa bersama dengan tim perpustakaan, Miss Pamela On, spesialis aplikasi CAS Scifinder dari Singapura. Untuk itu kami mengucapkan selamat bergabung dan terima kasih kepada seluruh sivitas Akademika Universitas Erlangga sebagai partisipan virtual workshop pada hari ini. Salah satu tujuan dari workshop ini kami selenggarakan adalah memberikan cara pencarian secara efektif dalam menemukan referensi yang relevan tentunya ya dengan cepat guna mendukung implementasi pelaksanaan Tridharma Perguruan Tinggi Sivitas Akademika Universitas Erlangga. Bapak, Ibu, para dosen, guru besar, dan mahasiswa di lingkungan Universitas Erlangga tentunya ya. CAS Scifinder ini kita langgan mulai tahun 2022 ini atas usulan dari Fakultas Sains dan Teknologi. Harapan kami tentunya database ini dapat dimanfaatkan semaksimal mungkin, sehingga tidak sayang kita setiap tahun ya mengalokasikan dana ratusan juta untuk mengadakannya. Bapak Ibu, ini pesan dari universitas. Jika pemanfaatan dari database e-jurnal yang kita langgan, yang kita adakan, itu terlepas dari kebutuhan usulan permohonan dari fakultas. Jika pemanfaatannya kurang maksimal, kita diminta untuk memutus langganan database tersebut. Itu kalau pemanfaatannya rendah dan tidak signifikan dengan dana anggaran yang kita keluarkan. Untuk itu Bapak Ibu ya, para guru besar, para dosen, mahasiswa Universitas Erlangga, tolong database CAS ini salah satu yang kita adakan dan juga merupakan permohonan dari Bapak Ibu ya, monggo dimanfaatkan sebaik-baiknya dan seoptimal mungkin. Dan Bapak Ibu Sivitas Akademika Unair ya, perlu kami sampaikan, dan ini nantinya kayaknya dipresentasikan oleh tim Latbang ya, mulai tahun 2022 ini untuk mengakses database e-jurnal yang dilanggan oleh Universitas Airlangga, selain bisa diakses melalui IP UNER, dimanapun, kapanpun, Bapak, Ibu, para mahasiswa bisa mengakses bisa melalui VPN. Untuk itu, monggo Bapak Ibu registrasi ke VPN ini. VPN ini merupakan salah satu software ya yang dibuatkan oleh Direktorat Sistem Informasi ya sebagai pengganti remote access yang kita langgan. Ini juga melanggan di Mbak Wiwi nih, Mbak Wiwi ya. Ini Mbak Wiwi remote access kenapa kita putus untuk tahun 2022 karena ini banyak masih banyak komplain dari dosen ya maupun mahasiswa terkait dengan pada saat login. Mereka itu menginginkan loginnya itu sama dengan login Cyber Campus, sehingga kami berkoordinasi ya, konsultasi ke Direktorat Sistem Informasi dan Digitalisasi Universitas Airlangga, dan VPN ini salah satu solusi ya yang dibuatkan oleh Direktorat Sistem Informasi dan Digitalisasi UNER sebagai pengganti remote access, karena untuk masuk ke sana itu menggunakan login Cyber Campus yang sudah dimiliki oleh para sivitas akademika di lingkungan Universitas Airlangga. Demikian Bapak Ibu yang bisa saya sampaikan. Sekali lagi saya mengucapkan terima kasih kepada Miss Pamela On dan Mbak Wiwi yang memfasilitasi acara ini, dan semoga acara ini dapat memberikan manfaat bagi Bapak Ibu sekalian guna mendukung sekali lagi implementasi program Tridharma perguruan tinggi, diantaranya yang sekarang ini merupakan target bagi dosen-dosen di fakultas terkait dengan penulisan karya ilmiahnya, publikasi, dan sebagainya, sebagai salah satu kebutuhan atau indikator untuk pemeringkatan Universitas Airlangga berkelas dunia. Demikian Bapak Ibu sekalian, kurang lebihnya kami ucapkan terima kasih dan mohon maaf jika terdapat kekurangannya. Akhirul kalam, billahi taufiq wal hidayah, waridawal inayah, wassalamualaikum warahmatullahi wabarakatuh. Monggo kami kembalikan ke Mbak Safti.
Ya, waalaikumsalam warahmatullahi wabarakatuh. Baik, terima kasih Ibu Suherni atas sambutan yang disampaikan. Untuk acara selanjutnya adalah tutorial OpenVPN yang akan disampaikan oleh Saudari Indah Rahma Cahyani, yang nanti akan menyampaikan terkait tata cara pendaftaran CAS Scifinder pada halaman website e-resources perpustakaan UNER. Kepada Saudari Indah Rahma Cahyani kami silakan.
Baik, terima kasih Mbak Sifti. Assalamualaikum warahmatullahi wabarakatuh. Good morning everyone. Good morning Ms. Pamela and Mr. Adran and Mbak Wiwi. So happy to see you again today. Baik, Bapak Ibu, sebelum nanti kita pada sesi untuk CAS Finder, bagaimana Ms. Pamela akan memberikan tutorialnya dan trainingnya begitu kepada Bapak Ibu serta teman-teman mahasiswa. Sebelumnya nanti akan coba kita jelaskan di divisi pelatihan dan pengembangan terkait dengan cara akses yang baru yang kita reminding lagi kepada Bapak Ibu untuk cara aksesnya. Sudah tidak lagi menggunakan remote text tapi move ke OpenVPN sehingga nantinya hari ini bisa tersosialisasikan dengan baik. Begitu nggih, saya izin share screen nggih Bapak Ibu. Baik Bapak Ibu, perlu kita jelaskan sekali lagi nggih bahwa jika dahulu kita menggunakan remote access sebagai cara akses utama untuk resources yang kita langgan, termasuk di dalamnya adalah CAS Scifinder, saat ini kita menggunakan OpenVPN sebagai cara akses utama. Begitu. Jadi ini berfungsi ketika Bapak Ibu tidak berada di kampus atau sedang berada di luar jaringan kampus, even Bapak Ibu berada di luar jaringan kampus, Bapak Ibu tetap bisa mengakses CAS Scifinder dari rumah atau dari mana saja Bapak Ibu berada, yang dimana OpenVPN ini berfungsi menjadikan seolah-olah kita berada di jaringan Universitas Airlangga, seperti itu. Nah, yang pertama yang harus kita lakukan, Bapak Ibu nanti akan sering visit ke laman Lib Unar AJ.id. Ini adalah laman utamanya kita. Kalau kita reload sebentar, nanti akan ada pop-up yang tertampil seperti ini karena masih dalam sosialisasi begitu. Di sini bisa Bapak Ibu klik pop-up-nya, kemudian nanti ada laman baru, tampilan laman awal begitu. Nah, di sini ada akses e-resources Universitas Airlangga menggunakan OpenVPN atau install OpenVPN. Nah, di sini kalau kita scroll down ke bawah, nah di sini Bapak Ibu ada halaman lain yang akan kita explore nanti. Nah, yang pertama adalah kita klik install OpenVPN. Kemudian kita akan lihat ada little describe di sini ya. Kemudian ada infografis juga. Kemudian ada petunjuk instalasi secara tekstual. Kemudian ada video tutorial instalasi dari YouTube kita di UNER Library, begitu nanti akan didirect ke sana jika Bapak Ibu lebih senang menggunakan tutorial by video. Jadi demo yang akan saya lakukan hari ini mengenai instalasi OpenVPN sama dengan petunjuk instalasi dan juga video tutorial yang sudah kita serve pada Bapak Ibu. Begitu. Kemudian di sini yang pertama nanti yang harus kita lakukan adalah Bapak Ibu masuk ke laman CAP Unair yang ada di tengah ini, untuk mengambil aplikasinya, kemudian kita pasang di PC kita. Kita klik di sini ada CAP Unair AJ.id, kemudian Bapak Ibu memasukkan atau teman-teman mahasiswa memasukkan NIM untuk teman-teman mahasiswa, kemudian Bapak Ibu dosen bisa memasukkan NIP atau NIK-nya gitu di sini. Kemudian ini diisi password cyber kampus, begitu nggih. Nah, di sini kemudian kita login. Ini adalah tampilan awal di laman CAP, begitu nggih. Nanti akan mungkin sedikit berbeda antara mahasiswa tendik dan juga lecture. Jadi di sini ada aktivasi VPN, simbol OpenVPN ini kita klik, nggih, Bapak Ibu, teman-teman mahasiswa. Di sini tertampil bahwa saya sudah terdaftar karena sudah berkali-kali untuk kegiatan sosialisasi, nggih. Nanti yang akan Bapak Ibu temui ketika belum sama sekali menginstal, begitu nggih. Ini untuk kita sebagai bahan pengingat saja nggih, bagi yang sudah menginstal kita coba ingatkan untuk teman-teman yang belum, begitu. Akan ada request VPN di sini nggih, Bapak Ibu, kalau belum sama sekali. Nah, di sini akan ada lisensi agreement yang harus kita centang, kemudian kita klik agree atau setuju. Nah, di sini akan tertampil laman ini, file yang diperlukan itu adalah poin 1, kita klik yang berwarna biru. Kemudian kita selesaikan satu dulu ya. Kalau Bapak Ibu menggunakan Mac, bisa diklik Mac di sini, ada Linux dan juga Android. Kemudian kalau di sini kita pakai Windows, kita Bapak Ibu klik yang biru tua ini, download OpenVPN Connect for Windows. Nah, di sini diklik. Kita tunggu sampai proses instalasi selesai. Kalau di sebelah kiri bawah ini ya. Nah, ini sudah ada, kemudian nanti apabila ada kendala, misalnya harus di versi Windows-nya itu harus di downgrade, misalnya, kita dari versi yang sebelum ini, begitu, nanti Bapak Ibu bisa ke Installation Instruction Alternative Version. Nah, di sini. Oke, Bapak Ibu bisa klik di sini 1, 2, atau 3, nanti bisa disesuaikan dengan kondisi Windows di PC-nya. Begitu. Nah, setelah ini selesai, kita kembali ke laman CAP, kemudian kita unduh lagi yang file poin 2, teman-teman, Bapak Ibu. Kita klik config OpenVPN. Nah, di sini sudah mulai terdownload Nah, yang kita butuhkan adalah dua file ini untuk memastikan bahwa OpenVPN akan kita pasang di PC kita. Begitu, nggih. Kemudian biasanya nanti akan berbeda-beda, nggih. Ada yang langsung muncul di laman download ini atau muncul di laman unduhan begitu atau di folder unduhan. Nah, seperti ini juga muncul. Tetapi kita coba yang dari browser, nggih. Kita klik. Kemudian kita prepare untuk install. Kita next, nggih, akan ada kotak dialog seperti ini. Kita next, kemudian kita accept, ya, kita install, kita tunggu. Nah, kadang Bapak Ibu di proses ini, installing ini akan ada kotak dialog yang akan muncul di beberapa PC atau di beberapa versi Windows. Nanti Bapak Ibu bisa klik yes atau OK. Baik, ini sudah finish, nggih. Nah, tandanya kalau sudah finish dia langsung terdisplay di laman desktop awal kita, ada simbol aplikasi OpenVPN Connect. Nah, akan muncul juga laman kotak dialog dari OpenVPN. Yang harus kita lakukan adalah kita klik kurung siku yang ada di tengah ini. Kita klik sampai 4 kali kalau tidak salah. Kemudian kita agree. Kemudian kita OK, kemudian kita klik file, no URL, kita biarkan saja, kita klik file, kemudian yang akan kita drag ke sini atau kita browse di sini adalah config yang poin 2 tadi. Jadi mengapa poin 2 tadi juga harus diinstal, Kemudian kita klik open, yang harus kita masukkan di sini adalah username kita diisi dengan NIM atau NIK atau NIP Bapak Ibu atau teman-teman mahasiswa. Kemudian kita save password. Passwordnya yang diisikan adalah password cybercampus. Kita klik connect. Nah, ini sudah posisi connected, Bapak Ibu. Kalau sudah posisi connected ini, kita akan kemana? Begitu, nggih. Nah, nanti tampilannya sudah kita create laman yang lebih easy untuk Bapak Ibu bisa menggunakannya. Nah, ini posisi sudah connected seperti ini. Nanti kalau sudah proses menjelajah ya atau sudah mengeksplor resources atau sudah setelah selesai mengakses CAS Finder, kemudian Bapak Ibu bisa mendiskonekkan kembali. Nah, ini didisconnect kemudian confirm supaya data kita, ada beberapa data kita supaya tidak terlihat terkait dengan privacy dan lain-lain begitu, Ge. Nah, karena ini saya berada di kantor, saya menggunakan jaringan Universitas Airlangga, jadi saya tidak perlu untuk menggunakan OpenVPN begitu. Ini berlaku ketika kita berada di luar kampus gitu, Ge. Lebih utamanya seperti itu. Kemudian kita kemana setelah sudah connected begitu ya. Nah, Bapak Ibu bisa kembali ke laman yang tadi ini, kembali ke home. Atau kalau misalnya masuknya dari website library begitu, Bapak Ibu juga bisa masuk ke e-journal databases, ini cukup disentuh saja. Kemudian klik link e-resources, tampilannya sama. Nah, kemudian kalau kita scroll down ke bawah, ya, Bapak Ibu, ini bukan satu halaman, tetapi ada halaman lain yang kalau kita scroll down ke bawah, nanti Bapak Ibu bisa temukan CAS di sini. Nah, ini Bapak Ibu, ada CAS Finder, kita masukkan ke list e-resources, ya, ada CAS Finder, kemudian Bapak Ibu bisa register. Nah, di sini. Nah, kemudian Bapak Ibu bisa mengikuti alur yang selanjutnya, dan itu nanti juga bisa masuk pada materi dari Ms. Pamela nanti. Jadi di sini saya tunjukkan, begitu nggih, Bapak Ibu saya antar, ketika sudah connected, Bapak Ibu bisa langsung register dulu, begitu. Kalau misalnya belum register, kalau sudah misalnya Bapak Ibu bisa langsung klik icon CAS Finder yang sudah kita plot di sini, begitu. Nah, kemudian mungkin sebelum nanti ada pertanyaan, biasanya ada pertanyaan yang sering kali ditanyakan terkait dengan akses e-resources adalah ketika time out, begitu ya. Ketika kita mengkonekkan time out begitu. Itu nanti kita bisa follow up langsung ke DSI beberapa kali seperti itu. Nanti Bapak Ibu bisa mengirimkan NIM ke kami, kemudian kita follow up oleh tim DSI pusat. Biasanya tidak sampai lama begitu ya, hanya beberapa jam bisa terkonek kembali begitu. Kemudian pertanyaan yang lain juga misalnya versi-versi begitu ya, versi Windows-nya tidak kompatibel dengan aplikasinya, nanti Bapak Ibu bisa langsung memfollow up ke kami di divisi pelatihan dan pengembangan. Ada saya dan juga Bu Nurma, nanti bisa ready on call untuk Bapak Ibu bisa menjelaskan apa yang menjadi masalah terkait cara aksesnya. Begitu. Baik, mungkin di sini saya bisa menerima 1 atau 2 pertanyaan. Mungkin selebihnya kalau pertanyaannya nanti panjang, nanti kita bisa follow up setelah acara begitu ya, supaya nanti bisa waktunya lebih panjang dan kita bisa selesaikan masalah Bapak Ibu terkait cara akses. Oke. Baik, Bapak Ibu, tidak ada pertanyaan. Kalau misalnya tidak ada pertanyaan, akan saya kembalikan ke Mbak Safety untuk beralih ke section berikutnya. Begitu.
Ya, baik. Terima kasih Mbak Indah Rahma untuk terkait informasi yang telah disampaikan. Untuk acara selanjutnya adalah dari Miss Pamela. Beliau dari Customer Success Senior Specialist CAS Scifinder yang akan didampingi oleh Mbak Wiwi Wijayanti. Kami persilahkan.
Ya, terima kasih Mbak Safety. Bisa dengar suara saya jelas?
Sudah.
Baik.
Ya.
Assalamualaikum warahmatullahi wabarakatuh. Salam sejahtera, namo buddhaya, om swastiastu kepada Bapak Ibu sekalian. Sebelumnya saya izin dulu untuk sesi ini kita akan menggunakan bahasa Inggris. Good morning, ladies and gentlemen. Greetings to all of us. Let me introduce myself first. I am Wiwi Wiyanti. I am from I Group Asia Pacific. I Group is representative size vendor and in Indonesia, and I would like to be your moderator today. Welcome to virtual workshop Size Vendor. Size Vendor and Airlangga University Library are hosting an online workshops and training for researchers and students to learn how to update your research by using Scifinder N. Before we start, I would like to say thank you for Ibu Suhernik, Ibu Profesor Nanik, Ibu Indah, Ibu Nurma, Ibu Ani, and all the team from UNER. Thank you for all the support. And also thank you for all participants who participated in this workshop. And greetings for Mr. Adran Ling as Customer Success Manager at SCS International. And to our speaker, Ms. Pamela On, as Application Specialist Size Vendor in Southeast Asia. Pamela is responsible for providing advanced training on Scifinder and STN to customers in Southeast Asia, Hong Kong, and Macau. Pamela has extensive experience as a synthetic and medicinal chemist. Prior to joining SAS, she was a senior scientist at GlaxoSmithKline, working on neuroscience drug discovery from hit to lead to preclinical phase. Pamela graduated from the Department of Chemistry at National University of Singapore with BSc Hons and MSc Chemistry. For today's special, because participants come from variety faculty, so if you never heard about Scifinder, what is Scifinder? I will give you a short brief explanation what is Scifinder. Scifinder is the most comprehensive database for the chemical literature. It's a core research tool for chemistry, biochemistry, chemical engineering, material science, nanotechnology, physics, environmental science, and engineering disciplines. And for more, Ms. Pamela will expand in her presentation. And if you have any questions, after presentations, there will be a Q&A session. So please don't be hesitate to send your questions by raise your hand, or you can unmute yourself, talk and asking Pamela directly, or you can send questions by message in chat box. Message can be in Bahasa or in English. I will help you to translate. And today, we will have giveaway. Pamela will give you 3 questions, and for the winner is will choose who participant answer fast and correct. So we expect all the participants can be more active in this workshop. Now let's begin. Dear Pamela, the floor is yours.
Thank you, Vivi. Thank you, everyone. Very good morning. It's a real pleasure to meet everyone. So let me share my screen. So as Vivi mentioned, we'll have sort of like a mini pop quiz throughout the session. So if you have not registered for a Scifinder and account, please do so following the very clear instructions that was shared earlier by Bu Inda. Okay, so get your account registered because some of the questions will require you to do some simple searches on the platform. All right, so I'll be asking the questions throughout the session. So just get registered and get ready. All right, so I will briefly go through the registration steps. Okay, so you should be able to see my PowerPoint slide. So if you click on the register for Scifinder and link on the library website, as you can see earlier on, you'll see this page and then click on next, and then it will ask you to accept the license agreement. Okay, so accept it, and then you'll be asked to fill in some information. So fill in the information as required. Just remember to use the university email address, all right, to fill in the sections asking for your email address. Then you'll be asked to also create your own username and password. So there is some requirements in terms of the password, how many characters, if you need special symbols, things like that. If you're not sure about it, you can click on the tips link and it will open up this little, I think it will open up in a new page. Okay, and then it will have the clear guidelines on what are the requirements. Once you have set up your username and password, as well as the security question, you can click OK or register, and it will be almost done. Okay, because there is one more step. You will need to then check your university email inbox for a confirmation email from CAS. And in that email, it will come with a confirmation link. So all you need to do is just click on the link and you will complete the registration. All right. So this is just a one-time registration. Once you have your username and password, you'll be able to log on to Scifinder and to use. All right. So if you have not already done so, I would highly encourage you to do it now. So like I say, later on, during the training, we will be asking you to do some simple searches on the platform. So, you need your username and password ready to access Scifinder N. Okay. Now, before I start the live demonstration, I will just go through a couple of slides regarding Scifinder N. And also, if you have any questions with the registration or if you encounter any difficulties, please let us know through the chat box, okay? So Vivi has already very kindly give a very nice summary of Scifinder N, so I'll just add on with a little bit more details. So CAS is part of the American Chemical Society. And what we do at CAS is to curate scientific content. So when we say scientific content, it could be your journal articles, it could be patent documents. We also have some conference proceedings and dissertations. So in terms of the scientific disciplines that CAS covers, we have up to 80 different sections. And the details of those sections can be found on the CAS website, but I'm showing you here some examples of the scientific disciplines that we cover. So it ranges from the more life sciences, biochemistry fields to the traditional chemistry fields and all the way to the more engineering disciplines. So we have a very wide scientific coverage. In addition, in Scifinder N, you will also be able to search the Medline reference database. So you have two sources of information to search using the Scifinder N platform. Okay. And one of the key points that differentiates Scifinder N from other literature resources is that at CAS, we have a team of scientists who curate and analyze the information that we obtain from the scientific public domain. All right, so as we receive the full text, whether is it a journal article or a patent document from the various publishers or patent authorities, the CAS scientists will read through the entire document. They would extract out the important topics that are discussed in each paper, any important compounds, and compounds could be your proteins, biomolecules, sequences, or your organic compounds, your inorganic compounds. All right, so a wide range of substance classes. Any important chemical reactions as well is captured. So all this information is added into Scifinder N. So you can actually search for specific proteins, specific metals, and find the key literature associated to it. Okay. So here I'm showing you the different types of information that you can find in Scifinder N. So I've mentioned substances, references, reactions. There is also chemical supplier information, regulatory information in Scifinder N as well. And more recently, we have added in the capabilities to search biosequences. So like the blast search that you can do on the NCBI website. So in today's session, we will focus on searching three types of information, which is references, substances, and biosequences. In the previous training session that we did back in March, all right, that was more focused on chemistry related topics. So we did a lot of learning on searching chemical structures, chemical reactions. All right, so I won't be covering this topic for today. Okay, but definitely in the future we can always repeat the same more chemistry focused trainings for you. All right, so let's look at substances, references, and biosequences. So I'm going to switch over to the platform. Okay, let me just take a quick look at the chat box to see if there are any questions. All right, okay, so there is the registration link for Scifinder N pasted in the chat box, so you can click on it to get yourself an account. Okay, so I'm going to switch over to our platform. Okay, so if you have your account ready, right, your username and password, you can log on and you can search along with me as I go through the demonstration. All right, so once you log on, this will be the landing page that you see. On the left-hand side is the different search types. All right, so we're going to look at substances, references, and biosequences. So let's start with the first topic of today, which is reference searching. So I'm going to switch over. So reference searching is, of course, putting in, typically you will put in your topic of interest, right? Put in some keywords, and then you'll find the related literature. So this is where you can enter your search query. There are other ways to search for references, such as author name. And if you click on the drop-down list here, you will actually find many other ways to start a reference search. Even using a substance name, a chemical name, you can also do a reference search with that. And you can use the plus sign here to actually run a search on multiple search fields or multiple starting points. So you can combine author name search with publication year, with organization name, etc. Okay, and under the end drop-down list, you have the different Boolean operators to construct, to link up your multiple search terms. So you can link up to 50 different search terms in a single search. But I'll leave that for you to explore on your own. All right, we will start with a simple topic search. Okay, so I'm going to type in my topic in the box, and we're going to look for references related to this particular type of bacteria. So I'm going to type in, okay, maybe I'll zoom in a little bit. So in case you like to see what I'm typing, so it's Pseudomonas. All right. And you can see as I'm typing, we do get some word suggestions. All right, and you can use that to help you double check your spelling, like what I just did earlier on. I was missing a D, or you can select the terms you see from the drop-down list and include it in your search query. Okay, so I'm just going to continue typing, and actually I found what I'm looking for. So I want to search for Pseudomonas, this particular type of bacteria. Okay, so I'm going to just click on it. I don't even have to type the full thing out. So I could definitely just use this and run the search directly. But there is an option to make this search a little bit more specific if you wish to. Okay, so in this case where we are searching the name of a bacteria, we are expecting these two words to always occur next to each other. So I can even include, okay, double quotation, just to make sure that all the references that I get in my results later on will definitely contain these two words, all right? And not only that, they are next to each other. So if I don't use any double quotation, what happens is that Scifinder will, of course, find all the references that contain these two words, but then it will also include some of the partial hits, right? Partial hits meaning references that maybe only contain Pseudomonas or only containing the second word here. Because sometimes partial hits can also be relevant, especially if you're working in a very new area, you don't have that many references. So you might want to also consider looking at literature that's working on a similar area, not exactly the same, but in an adjacent area. Okay, but if you want to be more specific, we just add in the double quotation. So let's run the search on this. Okay, so this is the number of results that I get. It's a large answer set, but not to worry, we will work to slowly narrowing it down later on. This answer set is currently sorted by relevance. Okay, but you can see there are many other ways to sort your answers. So the relevance ranking is very useful because it shows you the references that matches your query the best at the top. So this saves you a lot of browsing through large answer sets to find relevant literature. Okay, but of course you won't be seeing the most recent publication. As you can see, the first one here is actually 1983. So if you want to make use of the relevance ranking, all right, but to focus on literature that is more current, we can then use the publication year filter. So if I scroll down, you will see the publication year filter with a nice graph to show you kind of the publication trend, right, on this bacteria, which is, has been going up over the years. Let me just open this. So I can say, well, let's just focus on publications in the last 2 years, right? From 2021 to 2022, okay? So if I do it this way, I will have more recent literature, right? 2021, but it's still sorted to relevance. So I see the best matches first. And also, you notice there is a load more results button. So this, I would advise to use this only when your search query is specific, such as in this case, right? I have indicated clearly which are the words that I definitely want to see in my results. So in this case, I would actually load the entire answer set. If you do not use any Boolean operators, or if you do not use any of these symbols in your search query, I would advise not to load the entire results, because you will get a lot of what I mentioned earlier about the partial hits. Okay, so I've loaded the entire results. So we have here over 11,000 hits published between 2021 to 2022. All right. Okay, so we've looked at publication year filter. Let me scroll down and introduce to you other filter options that we can use to get this answer set narrower, more specific to what we're looking for. So a lot of the filters here are quite self-explanatory, like organization name, author name. I would want to introduce to you the filter options that are unique to CAS or unique to Scifinder, which is the concept filter. So let me click on view all to expand it. So earlier on in the introduction slides, I talk about the CAS scientists, right? Reading the full text, curating the information, extracting the information from the full text. So where do all this information go to? They would be in the database as concepts, okay? So over here, we have the full list of concepts from our 11,000 plus reference answer set. So concepts will represent the main research topics discussed in each of the papers in our answer set. And not surprisingly, because this is what we searched, right? So this, of course, comes in at the top. And then you see other terms that commonly occur in this area of research, like other names of other types of bacteria, antibacterial agents, and things like microbial sensitivity test, right? So these are all different aspects of this research area that people have investigated. So you can go through this list to see if your research focus, right, falls into any of the topics or concepts listed here. So this is listed, this is the most commonly occurring concepts, okay? So let me go through and pick something interesting to focus on. So maybe, for example, we want to find, let me see if I can find it. We want to find references that focus on cystic fibrosis, okay? So I can select this as a concept of interest, hit apply. So this would immediately narrow down our references to just over 373, okay? And all of these references will not only contain our bacteria of interest, right, or pathogen of interest, but in relation to the disease cystic fibrosis. Okay, so let's look at one of the examples in our hits. Let me scroll down to number 4. Okay, so this is a interesting one because you don't, well, actually I see it now, cystic fibrosis. Okay, so if you use the concept filter, the word cystic fibrosis doesn't have to appear in the title or the abstract, all right? Because what it is, how it is working is actually looking at the concept section. So I'm going to use this as an example. All right, we're going to open this in the new tab. You click on the title, open in a new tab. You will find the reference detail. Okay, so like I mentioned, the concept filter is actually working based on the key terms that are listed in this section, and we see cystic fibrosis listed down here. So don't be surprised to find references where cystic fibrosis is not mentioned in the title, it's not mentioned in the abstract, but it still is relevant, all right? Because like I say, we've done the indexing of the full text, and although it's not, this word is not mentioned in the title and the abstract, but in the full text, it is one of the important topics investigated. Okay, so concepts. The other area to look for the concept terms would be the Medline medical subject headings. We also see cystic fibrosis over here. So as I mentioned in Scifinder N, you search both Medline and the CAS reference database, right? So this is an example of a reference found in both databases, as you can see here. Okay, and Medline has their own way of indexing information. Therefore, there is the Medline medical subject heading. So this is added in by Medline. Okay, so we've also included this information so that when you run your search, it will really enhance your retrieval. Okay, because it can search in the concept section. It can also search in the Medline medical subject headings, as well as title and abstract, right? You can see the blue hit highlighting as well in these two sections. So the search query, your search query will have a lot of opportunities to find relevant references, okay? And then in this reference, yes, there is one more section here that I'd like to show you, which is substances, all right? So substances would be key compounds that are used in the paper that are important. So we mention and highlight them as well. And in some cases, you will see that there is this role information. So the role information just gives you additional idea how each of these compounds are utilized in the paper. So it says here biological study. So you kind of expect, have an expectation of how these substances are being used. If you find any of the substances listed here interesting, you can actually either click on the structure image, okay, and you will have this pop-up box that has additional links for you to run a further search on any of the key compounds. Even there's no structure available, you can still click on it and you still get this pop-up box. Okay, so you can find additional references. You can find commercial suppliers that sell beta-galactosidase, okay? Or you can look at substance detail of this compound. So let me just quickly show you. Okay, so this brings us to the substance detail page where you can find things like key physical properties, melting point, boiling point. There is an experimental properties tab where you can find additional experimental properties information and even experimental spectra. Okay, we'll talk about substance detail a little bit more later on. So let me just close this first. The other thing you can do is just, of course, click on the CAS registry number here directly, and you can also open up the substance detail page. Now, this particular substances, well, let me maybe backtrack a little bit. So you might be wondering why these two doesn't have structures, right? So this has a structure, this two doesn't have structures. And this is because these are large biomolecules. It's not possible to present their structure images in Scifinder and as an image, right? So there's no actual structure, but in this case, all right, so we've opened this up here. You see that this is an example of a protein peptide sequence. And if it's a protein peptide sequence, you can actually click expand on the sequence details, and you can see the sequence pattern of this particular protein. Okay, so this would be where you can get that information. And of course, from the substance detail, you can again branch out to find associated literature. Okay, so let us go back again to our reference. And of course, once you are happy with everything over here, if you like to view the full text, okay, in this case, I actually have access, I think, or maybe it's an open access. So let's click on it. All right, it will either say view source or view PDF. Okay, let's see. All right, let's see if I probably don't have access to this. Yeah, okay. So it brings you to the publisher's website. And through the identification of your university's IP address, right? If you are subscribed to this journal, you'll be able to view the full text. Okay, so for me, I don't have access to this, so I can't see the full text. But this would be the link to click to access full text if you are interested to view the full details. So let me go back to our answer set. open this up again. So other than kind of browsing through concepts to find terms of interest, you can also search for things you're interested in, because sometimes it's hard, right, to go through this, or even, to go through every single tab to find what you are looking for. So just search directly. So for example, if I want to look for references that focus on the 16S rRNA, I can just do 16S. All right, and then you can see here, anything that contains 16 or 16S is basically retrieved for us, and you can select the appropriate concept to further refine your references. So I'm going to select the 16S rRNA. Right, hit apply. Okay, so this is another set of results that focus on the 16S rRNA of this bacteria, this strain of bacteria. Okay, so earlier on we saw an example of a journal literature. So I'm going to take a look at something that is a pattern literature. I'm using the pattern document type. All right, so let's take a look at just this example here. So you notice for patterns, there is now an additional icon which we didn't see earlier on. So the pattern pack icon will allow us to access the full text pattern PDF by clicking on this link. All right, and if I go into the reference detail, okay, let me just close up these two first. We also have the pattern pack PDF here, or you can also click on this PDF here to view the full text. And in this case, let's open up the substances. Okay, we have concepts as well. All right, 16S rRNA. All right, and then in this case, the index substances are actually nucleotide sequences, right? So earlier on, we saw examples of references that are indexed with protein or amino acid sequences. This is a nucleotide sequence. And again, you notice there's the role information, and it even tells you that, okay, these are primers. Okay, these are used as probes. So you have, quite a lot of insights into this reference before going to view the full text. Okay. And of course, you can always click on the CAS registry number to view further details of any of these nucleotide sequences if you wish to. Now, if I go up again, all right, and just to show you the pattern pack option, right? So in this case, this is a Chinese pattern, so it's going to pull up the full text pattern. I'll just let this load, and we'll come back to it in a moment. All right, so let me just clear the document type. Okay, so just to show you, it links out to the full text pattern PDF. Okay, so other than the concept filter, another interesting or useful filter to use, scroll down further, is the search within results. So this is where you can put in your own keywords to refine answer sets further. So it doesn't have to be a concept, right? It can be your own choice of words. So for example, if I want to find references that use PCR to maybe sequence the 16S rRNA, right? So I can just type in PCR here, okay, run a search. All right, so you can see here, right, we get PCR, PCR. Okay, so the search within results works like the initial search query we run at the top. If you use search within, it will actually search in the title and the abstract. If you use the concept filter, it will just focus on the concept or the Medline medical subject headings. All right, so this is the slight difference between these two approaches. Okay, now that we have run our search, we have refined our answers, and we have a nice set of results that we are that is very relevant to our research area, you can definitely export them out if you need. Okay, so this is the download button, and there are different formats you can download your results. If you are using reference management software like EndNote, Mendeley, you can export them in the citation format. Okay. The other thing you may want to do is to set up an alert for this search strategy that you have just developed, so that anything new enters the database that's related to this bacteria and 16S rRNA and PCR, you will be informed via your email. Okay, so this is the button to use, save and alert. And you can save, to create an alert, you need to save the search. And when you're saving the search, you can also save the answers that come together with the search, all right? Or you can just choose to save the search itself, and then choose the appropriate alert frequency. So let me just put in pseudomonas, all right, 16S RNA, something like that, all right? Just remember to click save at the bottom. So whatever alert and save you have created is saved in this bell icon folder. So let me just open this up to show you. Okay, so this is what I've just saved. You can see that it saves the search together with the filters that we have used in our search. And you can always come in here to rerun the search if you need, and it will remember all the filters you have used, okay? And of course, you have the alerts set up for you as well. You can always change the frequency later on. Now, you only need to save the search if you are setting up an alert, or if your search has multiple filters applied and you want Scifinder to remember the filters. If it's just the first search that you run at the top here, this is automatically saved for you. We have here the history folder. So let me open this up as well. Okay, so you can see this is the search that I've just run. It's automatically saved for us. In fact, we have here the calendar. So you can actually, go back and recall searches that you have run months and months ago. So any of the days in the calendar that has a blue dot, those will represent days that you have used Scifinder in. Okay. All right, so let's go back here. Now, working on this topic, all right, so what if you want to run a search that contains multiple topics in a single search? Okay, so I'm still interested in this bacteria, but I want to immediately just search for maybe the use of this bacteria in bioremediation or bioadsorption, all right? Can I just combine everything in one search instead of doing it stepwise? The answer is, of course, yes. Okay, so for example, right, I'm interested in this bacteria, and then, like I said, using this for bioremediation or bioadsorption. Let me just type out, okay, bioremediation, bioadsorption. Okay, so how do we link up these multiple keywords? So definitely we won't use the double quotation because we are not expecting them to occur next to each other all the time. So in this case, I would use the AND operator, okay, to indicate to Scifinder that, I need this topic and I also need this other topic in my results. Now, bioremediation and bioadsorption, these are two things that I am interested in. It can either be bioremediation or bioadsorption. So in this case, I would then link up these two, okay, with the OR operator this way. All right, so it's very clear which are optional, which is compulsory in my results. All right, so let me run the search. All right, and now you can see in this case, there is the additional hit highlighting, right, that corresponds to the additional terms in our search query. All right, so like I say, if you don't see it here, it's probably in the concepts section, okay, bios option. All right, and I would load the entire answer set. Okay, so we have, quite a small answer set already because we have multiple search terms in this search. Now it's the time for my first question. Okay, so I run my first search and I get quite a nice answer set that's not too large, but I'd like to focus this even further. So like the first one, it's talking about cadmium bioremediation, but what I'm interested in is to find literature on the bioremediation of mercury. Okay, mercury. So can you type in the chat box, right? How would you do that to focus in the answer sets further, to focus on mercury? Okay, so I'm going to flash the question here. Okay, maybe let me just go into full screen. All right, so I've just run the first topic search, right, on Pseudomonas and then on bioremediation and bioabsorption. Now I want to focus it even further on mercury. So think about the filter options that we have discussed in the first example, right? How can you narrow the answer sets further? Type in the chat box, so the first person to get the correct answer will win the first prize. All right, so is it okay if I just show this screen instead of the full screen?
How many minutes you want to wait for the answer, Pamela?
Maybe 3 minutes.
Okay.
So technically you don't have to search. If you remember,
yeah, just type, yeah.
Yeah, type what's the option you will use. Anybody?
Bapak Ibu sekalian, boleh sekalian diisi siapa yang bisa menjawab paling cepat dalam chat box akan mendapatkan hadiah voucher, hadiah untuk setiap orang. Yang pertama voucher sebesar 100.000. Payung, kita ada souvenir, payung, agenda, dan goodie bag akan dikirimkan langsung ke alamat masing-masing pemenang. Dan pertanyaannya, how do you refine the results to focus on bioremediations or biosubstance of mercury? Bisa langsung diketik, Bapak Ibu, itu tadi caranya tadi sudah dijelaskan oleh Pamela. Kita akan tunggu 3 menit ya, berarti sekarang 11.07 sampai 11.10. We will wait until 11.10. 11.10.
Yeah.
Susanita, Ibu Susanita Utama, answer. Oh. Pamela, some answer, or this is just give her.
Sorry?
Their answer in chat box, but
okay, I see some. Yeah, all right. So so the first answer is from Susanita, right? So yeah, actually this is a possible way, and I think I should give this this answer as the correct answer, although I was thinking about something else, but this is definitely one approach. Okay, so Susanita is the first person. So congratulations.
Congratulations Ibu Susanita, mohon mengirimkan data dirinya nanti melalui chat box ke saya ataupun ke bisa ke tim UNER Ibu Mbak Sefti atau Mbak Indah.
Yeah. So
selamat Ibu Susanita, congratulations.
So let me go through this. All right. So I did get an earlier answer that asked me if it was running using the concept filter. Typically the concept filter is also a possible option, but actually I didn't try the. I'm assuming that mercury is not a concept. Okay, but I didn't actually check if it is. So let me just. Let's see. So mercury is a concept, but this is not the good approach in this case, because as you can see, it's only quite a few hits, but concept is one of the options to consider, all right? But like I say, not everything you want to refine with is necessarily a CAS concept. So you could always double check this section first. And if you don't find it there, it doesn't mean you can't refine with that word anymore, okay? So the answer I was looking for actually is the search within results, because you can type in mercury, and you can even have mercury or the chemical symbol of mercury, okay? So this is the answer I was looking for, but the answer provided in the chat box actually works the same way as the search within results. So this is a stepwise approach, okay? The other option is to, of course, just include mercury right at the beginning. Okay, so this would be the same effect as using the search within. So mercury, and if I want to follow what I typed here, I could do another or, so mercury or Hg. Okay, so which is why I think we should give that answer as the correct answer. Okay, so you have here mercury, occurring in the title and the abstract. All right, but the concept filter, like I said, do we have extra price? I feel like maybe I should also give the concept filter because it is a right approach to try. So maybe let's also give I think it was this. So the person DM'd me. DM'd me the answer, this. Let me see if I can find. Can you maybe type in the chat box so Vivi knows who you are?
Okay. We have three giveaway for 3 winners.
3. Okay, so we'll just do 2 questions then, since there's 1 prize left, right?
Okay.
Okay. Yeah. So, but this yeah, is the person still around? Ziz, are you there? You DM'd me.
What's the name?
I don't find the person. It just says ZIZ. Did the person leave?
And the person leave.
I can't find. So the person is not around anymore. Okay, never mind then, since she's not around.
Okay.
Yeah, okay, because yeah, I can't find the participant anymore. So yeah, I just wanted to say there was actually someone did send me a private message to ask if is it using the concept filter. All right, so the concept filter is also definitely one approach. Just that in this case, using the search within results or having it in the search query as end will give you a better retrieval than using the concept. Okay, so it's good example in searching literature, right? There's no one way to get there, and sometimes you need to trial and error, try different approach and see which one gives you the better retrieval. Okay, so thank you very much for the participation. So let's move on with this. All right, so now we have this answer set that is focused on mercury. I want to introduce the citation map feature to you, which is another way to find additional related literature. All right, so let's maybe use this as an example. So you can see here that this reference has been cited 119 times by other authors, right? If you open up the citation map, okay, I will, let me just, did it open up? Okay, let me click again. Citation map, yeah. This will allow you to see both the forward and the backward citations, right? So who cited this article and which references did this article cite? Okay, so for this, this information is not available. So let me pick another reference. Okay, let's go down to, I think this should have. Right, so this only has the citing. So let's see if this is, yeah. Okay, so this is an example that has both the cited by as well as the citing. Okay, so this is the root document, right? This is this reference. And within the citation map, we also have filter options, okay, including concepts. So especially if there are long lists of references on both sites, right? You can see here quite a lot of references. And to read through the abstracts for each one can be time consuming. So you can use the filter option to, again, narrow down to the area that you are interested in. Okay, and maybe I'll just pick like, let's say heavy metals. Okay, heavy metals, and then you can, okay, click on the titles to view the abstract of each of these references. So this would be the references over here, and the green references here would match the green circles over here. Okay, and you notice that when I hover my mouse over the circles, the references in this site will move accordingly. And not only that, there is the option to expand citations. So you can even go on to see, okay, this looks interesting. Let's see which other papers have cited this particular reference. So I click on expand citations and it shows me who cited this, and I can continue on for the references here. And it also works on this side. So I can also say, let's expand the citations in this direction. Okay, and then you can do it a few times as well. So this can really kind of help you see the whole citation, the full citation map on how many of these references are influenced by, you know, other references. Okay, so citation map. Okay, so let's move on to substance searching. So let me go to the home page. There's no questions, right, for reference searching. Okay. So, so hopefully it's clear, right, how to do a reference search and how you can link up multiple keywords in a search. And definitely, I think you have a very clear idea now how to, what are the options you have to filter down your answer sets further to get the most concise and relevant set of results, right? So the next thing we're going to look at is substance searching. So substance searching is, you know, typically you look for a compound or substance, and then you try to find either the associated literature, or you can use it to find things like properties. You can find experimental spectra, things like that. Okay, so we'll start with our first example. So I'm going to type in the name of this, I guess, enzyme, okay, carboxyl esterase. Okay. Again, there is the drop-down list, which you can use to double check your spellings. Okay. Sorry, I spelled wrongly, so I didn't find my compound. Should have used the drop-down list. All right, so it's carboxyl esterase. Okay, so we have here 5 results because it searches carboxyl esterase. This is what we're interested in, but it also searches the individual words in this name. So that's why we're hitting on this. All right, carboxyl. And then we also have here this sort of commercial compounds that contains carboxyl esterase as one of the components, and then there's other stuff mixed into it as well. So what we are interested in, the specific enzyme, right, would be this molecule over here, and we can click on the CAS registry number to view the substance detail. Okay, let's take a look. So in this case, again, there's no structure available, right? Because this is a huge, I guess, biomolecule, but this doesn't have the specific sequence. So it could be that the actual sequence for this carboxyl esterase is not known. So, it doesn't have the actual sequence code for this particular biomolecule. All right, but you have things like the experimental properties and experimental spectra. So if you compare this to the earlier compound, we saw this has a lot less information. So it really varies from compound to compound or substance to substance. And if you are interested in a property or a spectra and it says see full text, so it just means that you'll have to go to the literature source as indicated here, and you can find the links here to get the either crystal structure or molecular structure or for mass spec. All right, so if it's not clickable, right, you have to go to the literature source to find it. If it's available within Scifinder N, for the spectra, it will actually be a hyperlink that you can click on and view the spectra directly in Scifinder N. Okay, and then of course from here you can then link out to either references, reactions, or commercial suppliers. So let me just maybe link it out to show you. Okay, so this will bring us back to the reference answer set that we are looking at, and you can apply all the different approaches that we have covered to get this narrowed down further. Okay. The other way to search. So sometimes if your compound name, okay, if you're not sure, right, what is the right format? Okay, so let me show you an example of something that is quite long. Okay, so you can see some of these names are quite long, right? But let me just pick out one example. So again, I'm searching for another bacteria. Okay, so Helio. Right, so you can see some of these, they are really long. Okay, so if I'll just click one of them to show you. Okay, so so if you want to say, for example, find DNA nucleotide sequences related to a specific organism, okay, you can see that they are named this way and it's quite hard to know the exact name. So what you can incorporate into your search is the asterisk symbol. So we haven't talked about the asterisk symbol. So let me just get rid of this. Okay? So say you're interested in nucleotide sequences, so DNA. What I mean by the asterisk symbol is this, okay? Like a wildcard symbol. And you can also use this in the reference searching topic search, right? But you can also use this in substance name search. All right, so I can say I'm interested in nucleotide sequences for this, particular strain of bacteria. So I'm going to use this again. All right, Heliocobacter. All right, and then I have another asterisk. So you can use the asterisk in the middle of the name, or you can use it at the end. Okay, you just can't use it at the front. So this is a little tip, right, to help you find some of these biomolecules that can be quite tricky to find because you're not sure what's the exact naming format. So you can just put in parts of the name that will be in the name itself, all right, and then try to use this approach to find the actual substances that you're interested in. So I have here DNA and Helicobacter. Okay, and let's run this search. Oops, I think I got the spelling wrong somewhere. Okay, let me just go back. Okay, let's just check. Helicobacter. Yeah, should be correct. Okay.
Pam, I think it should be Helicobacter without the O.
Ah, yes.
Yeah.
Thanks. It's like thinking why I can't find it. Yeah. All right. Okay. All right, so this is what I mean. All right, so I have the word DNA, and then I have my species of interest, organism of interest, right, Helicobacter. So it helps me find over 19 results, right? So you have here, the DNA and the Helicobacter that matches the search term. So of course you can put in a little bit more, keywords, okay, to again narrow it down to maybe less than 19,000. All right, but just bear in mind this is an approach you can use to have asterisk symbol in the middle and asterisk symbol at the end to help you find compounds. Okay. So the other thing, yeah, let's open one to take a look. Okay. All right, and then here you have nucleotide sequence, and like I said before, you can see the full nucleotide sequence for this particular okay, I guess gene or something. And then you might be wondering, what is this related sequences? Okay, so and you may see this appear for protein sequences and nucleic acid sequences. So if there's related sequences, it's just telling you that there are other substances in our database that contains that have the same sequence detail or sequence pattern as this particular sequence we are seeing. So it's definitely worth checking out. It might be exactly the same, or it might be it has the same backbone but with some modifications. All right, so in any case, they'll be very similar to this, so it will be interesting to also explore the related sequences. Okay, and then finally, all right, so we've seen some examples to search like sort of biomolecules by name and search this sort of nucleotide sequences. The other search that you can do is, of course, just searching specific chemicals or compounds. Okay, so for example, I want to search lead 2+. Okay, so all right, so this would be searching a heavy metal. Okay. All right, and then we have here, this is our compound of interest, and then it gets all this compounds that contain lead as one of the components. All right, but this is the one that we are interested in. So let's take a look at, actually linking out to the references and then filtering it further. So I want to introduce to you one more filter, which we didn't get to see earlier on. So when you transition from a substance, all right, to a reference answer set, you now have this substance role filter option, okay? Or like in our previous example, if we have mercury, all right, in our search topic, that is a specific substance, right? You will have the substance role filter appear, okay? Or not, you won't see this. So don't be alarmed if you don't see this available, all right? You need to have a specific substance in your query, or if you cross over from a substance into references. Now, the substance role filter is, again, another way to help you filter down the references to the use of, in this case, lead, right? In the context that we are interested in. Because right now, when we cross over from lead to the references, right, it's 17,000 plus results, but they are anything and everything about lead 2+. Okay, so if I open this up, you will see the different roles available. So whether are you interested in lead being used as a pollutant or as a modifier, or maybe even as a catalyst, or are you interested in the use of lead in cosmetics, or maybe, I don't know, the industrial manufacture of lead 2+. So use all this, you can use all this to help you find the relevant literature. So I'm interested in the removal of lead from like water streams or whatever, right? So I can select this, hit apply. So now we'll just focus on references where the literature actually looks at removal of lead. So if you look at the title, it's kind of in that area already, right? Removal of copper and lead, high efficient removal of lead, lead to adsorption, etc. Okay, so you can see how by choosing this particular role, you have already focused your references to the area that you are interested in. And then of course, we can then combine this with, because it's still over 5,000 results, we can combine this even further and say, well, let's look at papers that use maybe ionic liquids to remove this lead 2+, okay? So I'll use the search within results and I say ionic liquid, okay? I'll put in the double quotation just to be a bit more specific. Okay, now I have not only lead, but also focusing on ionic liquid. So I only have 34 results now. So it's a small answer set, but it's very precise. So I'm just going to sort this by publication date, and we can have a look at a recent publication from Erlangga, okay, over here. So let's, let me open this up. We can see ionic liquid, right, highlighted. So this is quite easy to understand. So this removal or disposal, where did it happen? So for this, you'll have to check the substances section. Okay, and then you can see here lead 2+, and it has been assigned the role removal or disposal. Okay, so that's why when we use the substance role filter, it helps us to find this reference, right? That is essentially talking about adsorption of lead 2+. Okay. Then the other thing to mention is that there are actually different sort options. So it is probably advisable to first sort it as, I usually leave it as relevance, because especially when, you may get hits like this, right? Where it's kind of hitting on this sort of trait compounds where one of the components is lead. If you don't sort it by relevance, you may miss this and you think the compound doesn't exist, all right? So if you ever run a search and you find that the results look weird, just double check your sort option. So for example, if I change it to number of references ascending, okay, so you can see lead 2+ disappears, okay, because it's showing me, it is now at the bottom, it's showing me references that have the least, compounds that have least number of references, okay? Or if I sort by CAS registry number descending, so this will show me the larger CAS registry numbers first. So larger CAS registry numbers are usually the newer compounds. So lead 2+ will definitely appear at the end. All right, so it's good to just leave it as relevance first, and then you can sort it later on once you have confirmed the compound you're interested in exists in the database. Okay, so this is for substance searching. Now, before I move over to the last part, which is biosequence search, let's have another question. So actually, we're supposed to have 3 questions, right? But never mind, let me skip to question 3 first, and we can go back to the previous question again. Okay, so basically, is to do a substance search and then find the melting point of the substance above. So the first person to type in the correct answer, the melting point, will be our next winner. Okay. So for this, you'll have to actually use Scifinder N to get the answer.
Ya, Bapak Ibu sekalian yang sudah punya akun bisa langsung login, bisa langsung coba cari substance dari coenzyme Q10 ini, nanti bisa menjawab pertanyaan dari Pamela. Tadi Ibu Susanita sudah terima vouchernya, sudah langsung ditransfer, dan untuk hadiahnya nanti akan dikirim sore ini. So Pamela, Mr. Owen Muhammad, that answer from Bapak Owen Muhammad
Oh, okay. Let me take a look.
in chat box, but he said that he not using Scifinder.
Okay.
But if it's correct, I think, what do you think?
Okay, so I actually have another, the first answer is from somebody called Orange. Oh, keep
Okay.
myself private. Wait, so
Oh, please, everyone, if you don't mind, send the answer in everyone, chat box to everyone, because we can
so
see who is the first.
person prefers to keep themselves private, but okay, let me double check the answer first. So basically what you just need to do is, just copy this and right, search coenzyme Q10 in the substance box. Oh, I don't see it. Coenzyme Q10. Let me try again. So the the participants were able to find it, obviously, because they got the answer. All right, so I'm not sure what's going on. Let me just pull up my history because I did run the search earlier on.
Yeah, Pam, I actually found it. So it's actually 48 to 50.
Yeah, how come I can't find it? It's very strange.
It should reflect on this.
Yeah, I have no idea why, but oh yeah, okay. So I did run this earlier on. Should have 21 results. I got 19 earlier on. So yeah, I don't know what's going on. Yep.
Yeah.
Yes. So it's good that you guys found it so you didn't get confused. And Adrian, you did say right, 48.50. Yep. So so I don't know, do we want to give to the person that first sent the answer but in private, or do you think you want to give it to
I think can I know who's the name, because there also someone answer Bapak Muhammad Rizky Fadil.
yeah,
His answer also correct. Is
yes,
he?
yes, yeah.
Is he? But you choose the winner.
I choose okay, how about to be fair, we give it to the person who types it out in the chat box. Okay, because that was kind of
Okay.
the rules of the game. the chat box. So I'm so sorry, the person who teased me, but your answer is correct. So thank you for giving the answer. So for the last question, right? If you like to win the prize, let's put it in the group chat box. I think it's only fair so that everyone can see the answer and it's clear who actually typed in the answer first. Okay. So yeah.
Congratulations.
Yeah, congratulations.
Pak Muhammad Rizky Fadil.
Yes.
Muhammad Rizky Fadil, right?
Yes, yep. Correct. Yeah. But if you just want to participate for fun, like, but you want to be private and you just want to participate for fun, yeah, you can still DM me, okay? But for the prize, I think we'll give to people who put it in the public chat. Okay.
Yes, so for the next question, please answer in chat box to everyone.
Yeah. I think that's better. Okay. All right. So let's move to the last part of the webinar, okay, which is Biosequence Searching. Oh, before that, I did have an earlier question about the use of the asterisk. So this was also sent privately to me. So let me address that before I move to Biosequence Searching. So earlier on, maybe let me just retype it. I mentioned about the use of asterisk, right? So the asterisk is like the wildcard symbol. So basically, if, for example, I put DNA with an asterisk, I'm saying that I want to find all substances in the database that contains DNA as the first 3 letters. And then it can be anything else after that, all right? So this is the use of one asterisk at the end of your search term. So hopefully this search doesn't take too long. Then for the example that I used earlier on, I did a combination of 2 asterisks, okay? So you can see in this case, because I only have DNA and the asterisk, basically I find anything that has DNA at the front, and it can be anything else following the word DNA, all right? So it's like a truncation symbol or wildcard symbol. Now, if I use a combination of 2 asterisks, so my earlier example was Helicobacter, right? This time I get it right. And I have another asterisk. Okay, so this essentially is telling the system, help me find substances that contain DNA, and then it also contains Helicobacter. In between, it can be anything else. It can be just a parenthesis. It can be other alphabets, other names. All right, but I just need DNA, Helicobacter appearing somewhere in the middle of the name, and then it can be followed by other stuff behind. Okay, so that's why when I run this search, I can find things with DNA, Helicobacter, and then something else. Maybe to make it a little bit clearer, instead of searching Helicobacter, I'll put in this instead, all right? So I think it'll be a little bit clearer if I use this. Okay, DNA pylorus, then you will see, all right, so you can see this is DNA, in between DNA and pylorus, right, there's Helicobacter. Okay, and then after pylorus, it can be anything else again. So that's the use of the asterisk. So if you use this in reference searching, okay, so it's useful, like, for example, you want to search words like, let's say modified, right? The word modified, okay? But you also want to search things like modification, modifying, modifies, etc. Right? So if you ever have this type of situation, then you can also consider it using in reference searching. You can also do it this way. So if I put modi, maybe I can do modif, right? So I will be able to find modifier, modifies, modification, modified. Okay, so this is the use of the wildcard symbol or the asterisk. All right, so hopefully this is clearer now. So if you have any other further questions, please continue to put them into the chat box. Okay, so we'll move to the last section, which is Biosequence Search. All right, so this is where you can put in nucleotide sequences or protein sequences into the system to find similar sequences that match our sequence query. There are three types of biosequence search you can do on Scifinder N. So the blast search, for those who do biosequence searching very often, this you may be familiar with this, and this is what you can also actually do on the NCBI website. Okay. Of course, if you do it in Scifinder N, you have the additional sequences that are reported in patents, reported in literature that sometimes is not available in the NCBI database. Okay, then we have here the second type of search, CDR. So this is searching sequences based on the complementarity determining region of antibodies. Okay, so this CDR is searching a very specific type of I guess, biomolecules, which are the antibodies and the B cell receptors, okay, that section of biomolecules where you can search using the complementarity determining region. So the CDRs are the regions that bind with the antigens, right? So the antibody binds to the antigen. There's this specific, I guess, sequence code that, you know, binds with these antigens. So you can search using that information. And then the last one, Motif Search, is typically used to search shorter sequences. And you can do more specific type of sequence search. You can indicate exactly at different positions of the sequence, whether you want to allow some variation, things like that. All right, so let's look at 3 examples so you'll be able to kind of understand the difference of each of this search type. So like I say, the blast search would be the one that's most commonly used, and you can upload your sequence. So if you have your sequence search saved as a text file or FASTA file as shown here, you can just upload into the box here. Let's see, okay, documents. This is the old folder, let me. Okay, so I have here something that I have prepared. All right, so basically the file looks just like this. So I just have the sequence pasted out in text file. Okay, so I've uploaded this amino acid protein sequence. So I will indicate that this is a protein sequence, and I'd like to search for this in the protein database. And I'll also include the NCBI sequences, and I'm going to set, I'll just always set it to the maximum, which is 20,000. It says that it contains, does it contain? Yes. Okay. I didn't save this properly. Actually, up. My file, this is actually duplicate. Okay, so let me just remove this. Okay, let's just paste it in then. I'll get rid of this. So you see earlier on that it detects invalid characters. So it prompted me, and that's why I double checked. Okay, so this should be the sequence. And if you click on the advanced Biosequence Search portion here, it will allow you to vary some of the parameters of the blast search. So whatever settings you see here is kind of the recommended settings that is the NCBI recommends. Okay, but if you want to modify it to suit your research needs, research purposes, can do so. You can hover over the question mark, right? If you like to understand better what some of these parameters mean. Okay, and then we do have another default settings for shorter sequences. So if I click on this, adjust parameters, you can see that quite a few of the settings have changed. So this is optimized for shorter sequences. Again, by the NCBI guidelines, a shorter sequence will be anything that is less than 35 amino acid. Okay, so for me, this case, I have more than 35. So I'm just going to use the default settings for the larger sequences and just hit the search button. Okay, so once you've submitted your search, it will go into your history. So you can see that it is searching. It's usually quite fast, but you can always just leave it to run and then continue to do other searches and come back to check again. All right, so I've already pre-run this. So let me just double check the previous one. So I'm going to my search history and I'll open this. Okay, so this was run earlier on. So it's complete already. I'll just click on view results. Okay, so this is the layout that you will see. At the top will be your query sequence. Okay, so this is the first hit. The results are sorted by alignment identity. So the highest alignment, percentage alignment, and then going down. Okay, so this is the first hit. Query is the sequence that we have submitted for the search. Okay, and subject is the one of the hits that we found. All right, so in this case you can see that it is 100% alignment, right? And if you look at the alignment tab, okay, it's you don't see any pink highlighting, because if there is any misalignment, it will be shown in pink. Okay, and then there's also the subject tab, which tells you what is the CAS registry number of the first hit that we see. And this sequence is also found in NCBI. So you have the NCBI identifiers, and you can click on this, which will open up the NCBI website. And then finally, the last tab is to tell you which reference has this sequence been found in. All right, so this is the journal literature, and you can click on this reference button to pull out this reference. Okay, so let me just click on it. All right, so this is basically the reference that corresponds to this sequence. Now, if I scroll down, the second sequence, you can see that it is alignment of 96.64%. So there are some mismatches. Okay, and if you look at the alignment tab now, it shows you clearly where the misalignments occur, right? You have the query sequence and the subject sequence and the various positions where it doesn't match well in the alignment. The other thing to note is our query sequence is 283 sequence long. Okay, our subject sequence is actually over 1,000 sequence long, but the alignment occurs at one section of the larger sequence. Now, if you look at the numbers here, it will tell you more clearly. So the alignment actually starts at the 16th sequence of our query. All right, it's not sequence number 1. It's not the first amino acid, right? It's at the 16th position, and it's at the 798th position of our subject sequence. So this is how you tell where the alignment is occurring. Okay, and in this particular case, there is the subject tab, and you notice in this case, this doesn't have a CAS registry number. So if it doesn't have a CAS registry number, it means that this sequence doesn't actually exist in Scifinder N, but because we are searching the NCBI, some of the NCBI sequences, so you can still find it. So you need to click on the NCBI identifier to learn more about this sequence. All right, which also means this sequence is not found in any reference. So you can see there's click on the reference tab, and there's no reference tab here as well. Okay, could we mute the? Yeah, thank you. Okay, so so in this case, there's no associated literature for this sequence, but you can go to NCBI to find more information around the sequence. All right, then on the left-hand side, there are ways to filter the answer set further. So for example, you know, we don't really want to see, if you don't want to see results like this, where, you know, there is the alignment, it's 96.64, but this is only part of a much larger sequence, right? So in essence, it only overlaps here, but then there's a huge part that has nothing to do with our original query sequence. So you can use, in this case, you can use the subject coverage to get rid of this sort of, you know, larger sequence that are way larger than our query sequence. So I can say I want at least 70% of subject coverage, so that the sequences in the results are not too big, right? So they're not too different from my query. So I set it to 70. Okay, and then let's apply. So this would reduce the number of sequences All right, now it's only 50 results. And if I scroll down through the hits, you can see now the sequence length of our subject sequences. They are now pretty similar to our sequence query, right? So not super long sequences, okay? Then of course, there are other of these filter options you can play around to get you the type of answers you're looking for. So let me reset the filters again. The other thing I like to point out to you is, of course, we have the organism filter as well. So you can use this to say, well, I'm interested to only look at sequences from certain organisms, certain species. You can use this. Okay, so this is the BLAST search. Now let's look at the next type of sequence searching, which is CDR. So CDR, you notice that there are actually 3 queries to enter. So each of the query will represent a CDR region. So for this case, let me show you an example. You can of course paste in the sequence one by one, but if you want to upload the file, you need to save it as a FASTA file. So this is how it will look like. Let me open this up. Okay, so you just have to name it in this format, CDR1, CDR2 with the, this kind of arrow sign followed by CDR1. And then when I upload this, okay, let me just show you. It just goes directly into each individual sections, all right? And the same thing, I will maximize it to 20,000 and I'll run the Biosequence Search. So this time I'm not including the NCBI sequences. The same thing like before, it will go into our history, and in this case, this is pretty fast. So let's view the results. So for the CDR search, it'll be quite interesting because it has the Venn diagram. So it allows you to find, because we have 3 separate sequence queries, right? So you can choose to see results where it contains alignment for all the 3 sequence query that we have indicated in our search, or you can look at results that contain only 2 out of the 3 or just 1 out of the 3. Okay, for example, this Venn diagram. So obviously in the middle will be results that contain all of the 3 CDRs aligned along the sequence. You can choose to look at the ones that only have 2 or less. So I'm going to focus on the ones in the middle. Hit apply. In terms of the layout of the results, it's the same as what we saw for the BLAST search earlier on. Okay, you'll have the sequence query, and then you'll have the query sequence, and you have the subject sequence. It will show you the alignment. If there are any misalignments, it will be highlighted in pink. Then there will also be the subject tab, right, for you to see what's the CAS registry number or NCBI identifiers. And then there's also the reference tab to let you know what's the associated references where this sequence has been found. Okay, so you can see, and this is a little bit different because the alignment is found in 3 separate areas, right, rather than a continuous alignment. Okay, and then in this case, these are all the 100% alignment. Okay, and the same thing, right, subject and then references. So if you notice at the top, there is also a reference button, but this reference button, right, technically it should pull over all the sequences, all the references for this over 5,000 sequences over here. But at this moment, this reference button only pulls over the pattern literature. Okay, so you can use this, we call this global crossover reference button if you are only interested in pattern literature. If you're interested in non-pattern literature at the moment, you'll have to rely on the individual buttons. Okay, so let me show you what I mean. Open this in a new tab. Okay, so it brings over all the associated literature for the 5,000 plus sequences, but you can see it's all the pattern literature. Okay, so this is something that we are working to add in so that you can pull over both pattern and non-pattern literature. Okay, and then finally, the last biosequence search, which is the motif search. So like I say, motif search is typically for shorter sequences. And I'll just, use this as an example. Okay, so let's just start with this example and I'll start making modifications to it so you can see what is the advantage of motif over, let's say, blast search. Again, I have a protein sequence. I'll just set it to 2000 and I'll run the search. Okay, it's the same thing in the sense that you'll have the alignment, misalignment, just like in the blast search. So let me open up. Some of this. Okay, so if you don't put in any of the special symbols, the motif search works pretty much like a blast search, right? I find references where there is an alignment for my query sequence across another sequence. It can be longer than my query sequence. So if you are interested to find only sequences that contain the exact same number of sequence as my query, right, which is 11, I can use my subject coverage again. I can set it to 100%. Okay, and then hit apply. So this will allow you to find only sequences that have the same length as your query sequence. Okay. So with motif search, there are actually symbols that you can incorporate into your search query, such that you don't even need to run the search and then filter it by 100% subject coverage. So while this thing loads up, let me just rerun. Let's run the new search. Okay, so I'll go back to Biosequence. Yeah, it's still loading. So this was the motif sequence. So if I want to find exactly sequences that contain only this exact sequence pattern, I can put in the caret symbol at the front. Okay, followed by the $sign at the end. Okay, so if I run this search, it should only find this sequence that is exactly the same, this pattern and then this length. All right, so let's run the search. This is still loading, so hopefully it loads up soon. Okay, and with the motif search, I mentioned that you can also indicate variables, right? So working with this example again, if I have the caret symbol at the front, followed by $sign at the end. Where's my $sign? $sign. Suddenly can't find my $sign. Yeah, okay, over here. So I can say, well, at this first position, besides this amino acid, I'm also interested in, so I open up the square brackets. It can be an X, P. Okay, so these are the different amino acid codes. Okay, so this is a little bit more, I guess, complex, but you can have really a lot of control over the search. So I'm interested in sequences that are this long, but at the first position, it can be any of these 3 types of amino acid. Okay, so let me just submit this to run because it takes a while to load up the results. Oh, it's still loading. Okay. Let's see if me again pull up my search that I've run previously. So this is still running. This looks like it's done already. No, it's not. View results. Okay, let's just look at this. Sorry. Just look at this. Okay, which has the caret symbol as well as the alternate amino acids. Oh, okay, it's finally loaded, right? So what did I do with this? I think I limit it to 100% subject coverage. So when I limit it to 100 subject coverage, you can see I find sequences that are also 11 amino acid long. Okay, this will be the exact match. It's finding itself, right? But then also there are the ones that are a bit shorter, that it's also 100%. Okay, now if let's see if the other one is done. This is still not done. Now, if I'm sorry, it's taking a while to load. So we'll, okay. I don't have an example of this. Oh, it's done. Okay, let's open this up. This is also done. Let's open this up. Okay.
Pam, I think we still have 5 minutes left.
Yeah, okay, sorry. Yeah, basically with the sequence search, sometimes the results take a while to run and it can take a while to load. So maybe I think you've at least seen how you can run the BLAST search and the CDR search. Okay, so the motif search, it's I guess the key advantage of that is you can run specific sequences like this, where you can indicate the specific length for the results you want to see. And you can also have the individual have some variation. All right. So I think maybe I don't dwell further on this. It's also getting a little bit maybe too technical as well. So if there's interest to learn more about biosequence searching, we can always do separate session just focusing on that. So with the last 5 minutes, I don't know, I have one more question. Do you want to do that, or shall we just skip it?
Yes, we can.
Do the last question. Okay. So I have one more question earlier on. Okay. So it again requires you to do a quick search, and it's just to then find the publication from University of Erlangen on this topic. So I didn't specifically show this filter option in the demo earlier on, but should be easy to figure it out.
Yeah, so for all the participants, please kindly to find the topic Zoom fatigue or video conference fatigue from University of Erlangen in Scifinder M.
Yeah, probably you're feeling this right now, Zoom fatigue, 2 hours.
Yes. And please answer your questions. Please tell the answer in chat box to everyone not in the M. And for Mr. Muhammad Fadil, I have transferred the GoPay, and I think I already received.
Is anybody? I have one. Oh, okay, let's see. We have one answer. Okay, so I'm just going to do the search, all right? And really what you need to do is just go into the organization. You can see here, there's one from University of Erlangen. So this is what it's, yeah. So yes, that's the right answer.
Okay.
Okay.
Yeah, this he's the same winner
Okay.
from last questions.
All right.
Congratulations again.
Yes,
Pak
congratulations.
Muhammad Rizky Fadil.
Yeah.
Thank you for your active in this
Yes, thank you for your
workshop.
participation.
Okay, so
we
if
can.
you still can we still have any session question and answer time, Mbak Indah? Okay, if you still have any questions furthermore, maybe you can send by email to me or to Pamela directly, and we will try to answer. And Mr. Adran just sent the link related how to use Scifinder N for more training informations. So maybe if you still want to know about this training session, you can check in the link. So yeah, if no other questions, I think we can close this presentation, Pamela. You still have something you want to say?
No. So yeah, any questions later on, just let us know.
Okay. Once again, thank you, Pamela. So we just learning in Scifinder N about substance, concept, netlines, citations map, biosequences by BLAST, CDR, and Motif, find pattern literature from many references. And thank you for Pamela again for sharing this knowledge. And it's been my pleasure to be part of this event. Looking forward to see you in the next event. Last but not least, I wish you all have a pleasant day and stay safe and happy fasting. Yeah, Mbak Sifty, I give the time for you. Thank you.
Ya, baik. Terima kasih untuk Mrs. Pamela dan Mbak Wiwi Wijayanti atas penyampaian informasi terkait CAS Scifinder. Baik, kalau tidak ada pertanyaan dari teman-teman atau Bapak Ibu yang sudah hadir pada hari ini, mungkin saya bisa tutup acara saja, ya. Ya, baik. Thank you very much to Mrs. Pamela for explaining very details and clearly, and also thank you to Mr. Adran Lee. Dan terima kasih atas kehadiran Bapak, Ibu, dan Saudara telah mengikuti kegiatan workshop pada hari ini. Perpustakaan akan melakukan evaluasi terhadap tingkatnya keterpakaian database CAS Scifinder pada akhir tahun. Untuk itu, kami mengharapkan pada Bapak, Ibu, dan Saudara dapat memanfaatkan secara maksimal untuk database ini. Baik, sampai bertemu kembali pada acara berikutnya. Salam sehat selalu. Saya tutup. Wassalamualaikum warahmatullahi wabarakatuh.
Waalaikumsalam warahmatullahi wabarakatuh.
Thank you, Ms. Pamela. Thank you, Mr. Adran. Thank you, Mbak Wiwi.
Thank you.
Terima kasih semuanya.
Everyone. Thank you.
Thank you. Bye-bye.
Happy fasting.
Boleh saya tambahkan di sini, Mbak Sifty? Bapak Ibu, apabila ada terkendala dengan cara akses, bisa kita selesaikan hari ini, bisa raise hand, sehingga saya tahu siapa-siapa saja yang ingin kita clearkan hari ini terkait dengan cara akses? Baik, dari Pak Rosyid sudah berhasil nggih? Untuk teman-teman yang lain, Bapak, Ibu, dosen, teman-teman mahasiswa juga terkait cara akses, ini sangat penting nggih untuk ketika cara aksesnya berhasil, nanti kita bisa mudah untuk masuk ke CAS Finder-nya.